Guys, we are the Mitos. I'm David.
Riker.
Lorena. Lily. And we're going to be, Ivana. We're going to be going over mitosis and the mitotic spindle. And the first phase we're going to talk about is interphase. So during interphase, we actually start to get our the appearance of our centrosomes. These are going to be our centrosomes. And in our centrosomes, we have two centrioles, and the DNA itself is gonna and
This is a phase where we talked about earlier where DNA is replicating during interphase. And then after interphase, we have prophase, and that's where the chromosomes start to condense into the chromatids, and the nuclear membrane at this point starts to dissociate. I'll just take that out. Dissociate. And at this point, the chromosomes have replicated already. We didn't have that many, but there's usually 6 pairs here. And then also during prophase is when the two centrosomes start to move to their opposite ends.
so this is prophase, and we're just re-explaining. This is actually where the mitotic spindle begins to form, and as you see the sister chromatids right here, they're starting to break apart, and the nuclear membrane are broken apart, and they're condensing into smaller ones. During prometaphase, we actually get to where the microtubules are connecting. So here we have the purple one is going to be the kinetic core, and it's going to, this happens like very, really fast. And it's going to connect to this is the kinetochore microtubule, and it's going to connect to the kinetochore on the chromosomes. So everything's just kind of like getting in place, not really in place yet. The centrosomes themselves are being pulled away by the antimicrotubules, and we are also getting the polar microtubules. So he's going to go more in depth about what exactly this.
So what she had explained, right now we're in the prometaphase. It's happening really fast. So here, as you can see, we have 3 main microtubules. We have our So we have our polar microtubules. Oh no, actually, I'm sorry. The green ones are going to be our polar microtubules. The orange one is going to be our kinetochore microtubules, which is going to be wrapped around our sister chromatids, which allows the pooling. And then the purple ones at the ends right here are our aster microtubules. And then within, so these little white balls would be our spindles. And then inside the spindles are going to be the aster, or no, this centrioles, which is our 2 pairs. Yes. Yeah.
In our centrosome, we have 2 centrioles, and the way how to describe it is like there's 2 people driving a crane, and the crane itself would be all the different tubes, but inside would be the centriole, and the crane itself would be the centrosome.
And if you think about it too, it's kind of doing this, and then towards the end, it's going to go back to its last phase. But that's going to be
Metaphase, right?
Yes.
And we're going to move on to metaphase.
You guys are named. I can do it.
And during metaphase, this is where these are continuing to attach, and everything's starting to kind of like line up pretty. And this one is going to be attached to that tendon cord. And the big one, how we described it as the polar microtubule, that one's actually pushing away the sister chromatids. So this pink one would be pushing like the green one away from it. And the next phase is anaphase.
Anaphase. Okay, so the next phase is anaphase, where basically it just goes to opposite ends.
The tendon cord breaks apart, the centromere is exposed, and they're kind of just get pulled to opposite ends. So this is where, again, the tendon cord is breaking apart, still attached to the tendon cord tube, and these things are just kind of separated. That's and then telekinesis.
So now we have telophase. Telophase involves chromosomes that reach their respective poles, so they've pretty much completely separated out. They condense into a more nuclear membrane and separate into two more distinct cells or vacuoles. And as you can see, they're kind of condensing into two separate nuclei as well, slowly starting to. And there's also a small presence of the cleavage for. And then with cytokinesis, the nuclei are completely separated into two daughter cells. So these cells are pretty much completely separated. The only thing that's holding them together then would be the cleavage furrow. Now the cleavage furrow is basically just how the cells divide, but that's only in animals. In plants, they also have something called the cell plate. I don't know if y'all remember that from biology or not. But basically, the cell plate actually divides a little bit differently. They divide by something called the the phragmoplast, and it's basically composed of microtubules that push against each other until they fully form two separate cells.
And during the telophase, that's when these are already pulled apart, and the mitotic spindle actually disappears, and we get the nuclear membrane to separate the two cells.
And that's pretty much your basic mitosis.